Today, we are pleased to present ExtraMIR, a joint initiative led by the International Dairy Federation in collaboration with the International Committee for Animal Recording. This project aims to intensify the use of Fourier-transform mid-infrared spectrometry by harmonizing and standardizing terminology, analytical procedures, and tools for quality assurance. Before introducing the application areas of this initiative, it is essential to first present the standard milk analysis process. Bulk or individual bovine milk samples are collected on farm or at a milk processing plant. A milk testing laboratory can then analyze the sample using Fourier-transform mid-infrared (FTMIR) spectrometry to determine the milk's fat and protein contents and sometimes other parameters. A box within the milk analyzer then generates the FTMIR spectrum for the corresponding milk sample. Please take a closer look at this box. First, a mid-infrared beam passes through the sample. The portion of light absorbed by the sample is then measured using a detector. This process is performed for at least 800 different frequencies, generating a spectrum. This spectrum, which results from the vibrations of specific chemical bonds at precise frequencies, represents the overall composition of the milk. However, to be useful, it is necessary to extract the most important information from this spectrum to quantify specific milk components, such as fat or protein. This is achieved using equations that provide, for example, the raw predictions of fat and protein content. These predictions may be influenced by infrared signal quality. To mitigate this, the spectra can be firstly corrected following manufacturer's recommendations, and accretion slope and bias can be optimized as part of the IDF/ICAR recommended routine quality controls to ensure prediction accuracy. The final predictions can then be exported to the farmer or processor for further assessment. To predict the various components in milk, different equations are required. Third-party and/or manufacturer's equations can be used depending on the manufacturer. Many other equations have been developed by various research teams and laboratories. This could prove valuable in enhancing the sustainability of milk production. By extracting and utilizing the spectral data more effectively, particularly through the creation of Fourier-transform mid-infrared (FTMIR) spectral database, new possibilities can be unlocked. It's time to think outside the box. The ExtraMIR proposal aims to facilitate the creation of FTMIR harmonized spectral databases by creating guidelines, empowering operators to evaluate the spectral robustness of their equations and to think outside the box. This initiative will support the milk testing laboratories to export FTMIR spectra from the analyzer, standardize them using a spectral correction process based on specific milk standards, and produce harmonized spectra. Once standardized, equations can be applied to these corrected spectra, delivering accurate and reliable final predictions to farmers or operators. To achieve this objective, ExtraMIR is structured into 4 work packages. These include the harmonization of the reference methods and values necessary for developing the equations, the creation of an independent validation set to compare equations developed for the same traits, the establishment of a quality control system for spectral data, and, importantly, a work package focused on communicating the benefits of new mid-infrared analysis to all stakeholders and guidelines. Let's think outside the box together. Join the project today if you haven't already.